Thank you so much, Jay, for the great introduction. And it was a great honor for me to participate in this session. And I hope that everyone posted this presentation will get a good sense about the landscape of pediatric cancer and also the progress we made in terms of developing innovative software tools and sharing data to advance pediatric cancer research. So I will start making sure that the slide look good. Okay, so I'll start with my discussion on genomic variants in pediatric cancer landscape, precision oncology, and data sharing ecosystem. So in the kind of the conventional world, what we consider genomic variants to precision medicine is a one-way direction, which starts with a research cohort, and we perform molecular profiling, including next generation sequencing on this cohort. And from the resulting data, we will identify prognostic markers or markers that predictive of drug sensitivity and resistance, or markers that predict of adverse events. And this can be applied in future applications of clinical samples. However, as clinical sequencing become a standard approach, we feel that this is actually an iterative process, that we can learn so much from the genomic data and the outcome from the patients. So the resulting data generated from the clinical profiling could also be feedback to research as a loop to launch another iteration of research that keep improving on the process of our effort in improving the care of the patients. So our journey on pediatric cancer genome analysis starts from the research prospect and with the launch of the Pediatric Cancer Genome Project in 2010, where we analyzed major high-risk subtypes of leukemia, solid tumor, and brain tumor. So for this effort, we have analyzed pair tumor whole genome sequencing data from 700 patients, whole exome sequencing of 1,500 patients, and over 1,000 RNA-seq. More importantly, we also performed experimental validation for over 2 million somatic alterations. And that ensures that we can develop innovative analytical tools that can support clinical investigation in the future. So the research outcomes of these studies have been published in 35 high-impact journals, defining a variety of pediatric cancer driver genes that can be used for developed clinical application. So once we complete the data analysis on the individual cancer types, we also collaborate with the National Cancer Institute on the target initiatives to perform pan-cancer analysis so that we can looking for the commonality and differences between different subtypes of pediatric cancer. So in this study, we have collaborated with leaders in ALL, AML, neuroblastoma, Wilms tumor, osteosarcoma. When we were launching our study, we also realized that Germany has also started with their own pan-cancer analysis. So to ensure that we coordinate this research effort, but without repeating, having redundancy, we actually supplied all the whole genome sequencing data from St. Jude-led PCGP to the German group, so that we can analyzing independent cohort and kind of cross-validate our findings with independently. So for the NCI target project, we have analyzed close to 1,700 pediatric cancer patients that were analyzed by whole genome, whole exome, or RNA-seq on these 6 different subtypes. So the whole genome sequencing by the target project uses the complete Genomics Platform. And this company actually produces about 100 million candidate variants due to some of the PCR artifacts involved in sequencing protocol. So what we have done is we have developed a very elaborative filtering and QC check processes, so that at the end, we were able to utilize about 44,000 curated variants from these 100 million at the start, and apply the statistical analysis tools to identify 142 driver genes. We also applied another approach that were initially developed for clinical sequencing initiative called pathogenicity classification. That led to the discovery of 82 additional variants that cannot be discovered by the statistical test. So what we learned from this data set is that the majority, 50%, 5% of the driver genes are absent in the 3 pan-cancer studies of adult cancer. And this really have a profound impact on how we should consider pediatric cancer treatment, that pediatric cancer patients cannot be treated merely as a small-sized adult. The other lesson we learned is about 62% of the driver variants are caused by structural variations or copy number alterations. This really indicating that the whole genome sequencing profile is a critical and important technology that had to be applied in both the research and clinical testing of pediatric cancer patients. So when we're looking into the biological processes that altered in these 6 subtypes of pediatric cancer, this is a map that shows how the processes on the y-axis and the cancer genomes, different subtypes on the x-axis. And we also mapped the proportion of the genes unique to pediatric cancer in these colors. So what we've shown here is that transcription factor, which is a top regulated, dysregulated class of the genes in pediatric cancer, the majority of these were absent in adult cancer, suggesting that therapeutical approaches targeting this group is required to ensure that we find new approaches to target these driver variants that are specific to pediatric cancer. We also performed the mutational signature analysis in pediatric cancer in collaboration with Vladimir Alexandrov from UCSD. So initially, we did not expect to see anything that of major surprise, but strikingly, we were finding a subset of the pediatric leukemia that harbor UV light signature in their genome. And as we can see here, these mutations caused by UV light signature actually account for a high proportion of mutations in their cancer genome. And we look into the details of the breakdown of the AL subtypes as well as ethnicity. We can see the majority of all patients are Caucasians or Asian. There's no African American descent. As well as they don't have kind of driver fusion genes normally discovered in defining the subtype of leukemia patients. So it indicates that perhaps UV exposure may be related to, may increase the risk of pediatric leukemia. So in this cohort, we just lack of sufficient number of the patients to make statistical calculation of UV association with the patient. So future analysis is required to replicate the study to establish the link of UV exposure to leukemia onset. And this study was multi-disciplinary research that with participation of two former postdoc fellow Yu Liu and a senior scientist Xiaotu Ma in my lab, with support from the NCI target team as well as variety of different laboratories in St. Jude, including the Genomics laboratory, the software development group, Biostats, and also other collaborators in the external collaborators like Vladimir and Bob Huether, a former trainee in my lab. and the study is supported by National Cancer Institute. So the next I want to talk about the collaboration we have with Shanghai Children's Medical Center with a focus on looking into drug-resistant mutations in relapsed pediatric ALL sample. And this is the study was recently published in Blood this year. And we have performed whole genome sequencing and RNA sequencing of diagnosis relapsed germline trials of 103 patients treated in China. And these patients can be divided into, based on the time of the relapse, into 3 groups of very early relapses, which relapse occurred before 9 months, early relapses between 9 to 36 months, and this is where before the end treatment of leukemia, or late relapse, which defines a subgroup of the patients that have has post-treatment relapse. So what we have discovered is that relapse-specific mutations are enriched in 12 genes, which are highlighted here, that known to be involved in pathways of the drugs that were used in treating leukemia patients. And these drugs include glucocorticoids, dalpurine, and methotrexate. And when we look at the leukemia, which patients group that these leukemia resistant mutation occur, we do see that very early relapse patients, meaning that during the treatment relapses, they have a much enriched mutational burden in these 12 genes compared with very early relapse, suggesting that acquisition of these mutations may be related to therapy. Again, we looked into the mutational signatures of that present in the diagnostic samples and the relapse samples of these patients. So in this graph, the y-axis representing the number of the mutations, somatic mutations, and each of these columns representing a paired patient in the diagnosis and relapse. So we can see that, and the color coding representing the mutations attributed by different mutational signature. So what we can see here is that at relapse, the mutation burden has increased substantially compared with the relapse. And also we found 2 novel signatures, what we call A and B, that were shown in blue and magenta color here, that are only present in relapsed patients. And we also see they're absent in the very early relapse, but were highly enriched in the early and late relapse, again suggesting a potential link to therapy. So these novel A and novel B signatures are not only providing passenger mutations, as we could see that some of the relapse-specific genes in the previous 12 gene panel, PS1, TP53, could be introduced by, caused by novel signature A, and NT5C2, NR3C1, and TP53 could be caused by novel signature B. So to find out which drugs might be related to the rise of these 2 mutational signatures, we performed this in silico experiment by looking into mutational signatures of the pan-cancer adult cohort analyzed by the PEACOCK groups, as well as mutational signatures in the NCR target, which include both diagnosis and the relapsed diseases in neuroblastoma, AML, and AL. So what we found is that normal signature A appears to be predominantly discovered in the Shanghai cohort. We only found 1 patient in ALL target that relapsed in this, that have this novel signature A, suggesting an agent that probably unique to the Shanghai treatment that led to the rise of this cohort. By contrast, novel signature B were found in both the AL target and relapses and Shanghai cohort. suggesting that the agent used in both the US and Shanghai cohort may contributing to this, this signature. So we speculated thiopurine treatment, which was used in the maintenance therapy post the induction therapy in AL treatment, may be responsible, given the timeline of acquisition of these mutations between the 9 months to 36 months in the patient cohort we received. So to prove that, what we have done is to perform an an in vitro experiment. So the first experiment we did is to use the REH cell line, which is a known leukemia cell line, and treat them with thiopurines, as we've shown here. So each of the columns representing one cell line with specific treatment. And we isolated single cell clones and perform whole genome sequencing on these post-treatment cell line. And the result is actually a little bit disappointing, as we found that most of the new mutations we observe appear to be in these single cell colonies that we isolated appear to be shared among these individual single cell clones, suggesting that they are pre-existing in the parental REH cell line. So the treatment is only selecting for single cell that will already harbor these mutations. So when we did a little bit further check, we recognized that AL cell lines, most of them were acquired from relapsed AL cells instead of the untreated primary cells. So that's indicating it is not the right model to looking into the acquisition of novel signatures that due to the treatment. So the normal breast cancer cell, breast cell line, MCF10, was previously used for confirming the acquisition of cisplatin signature in relapsed breast primary tumors. So in the second experiment, what we did is using the MCF10 cell line and treated with thiopurine, and also comparing the treated cells with untreated cells, as well as we also performed the cisplatin treatment in the MCF10 cell line. That way we could look into whether our experiment to use this cisplatin treatment as a positive control to see if we could replicate the existing result. And this time we do see very distinct private mutations in contrast to the REH, suggesting the experiment is work. So first, we were able to confirm our positive control experiment of cisplatin did reproduce the same signature as what was previously published as the cisplatin signature. And then when we look at the novel mutations gained from these two experiments, they indeed bear the same signature as we found in the novel signature B in the patient sample, confirming that treatment to thiopurine indeed caused the novel signature B that we observed in the patient sample. So this study really tells us that the cause for the relapses in AL will require expansion of existing models for relapse. So the current models that account for the relapse are two, which is one is called de novo resistance, meaning that the cells does not respond to drug treatment at all. And this was not observed in our ALL study because our patient went through remission. So the debulking of the tumor already happened in in our cohort. The second is chemo selection. And this is kind of the predominant model, which suggests that prevailing model that suggests that selection of the small single clone and leading to the relapse of the cancer. And this model, we believe, is account for very early relapses in our cohort. So the new model we propose is that there's actually a two-phased approach leading to relapse. First is a chemo selection of a persistent clone that can survive chemotherapy, as shown in these light blue line here. But chemotherapy will induce additional mutations that lead to the clonal expansion of relapse. And these are likely, the model is likely to account for the early and late relapses. So this really suggests there could be a model of for monitoring the persistent clone before their acquisition of chemo-resistant mutation that may determine that some of the chemotherapy need to be ceased and switching to some other for those that for patients that survived the initial chemotherapy. So the most of the computational analysis was led by Sam Brady in my laboratory. And there's multiple institutions from Shanghai, Tianjin, and Anhui in China participated in this study. And Ben Raphael's group from Princeton University helped with computational modeling and analysis of clonal evolution in this study. Now I want to switch gears, talking about kind of my changes from research to clinical genomics. So this is the effort that requires collaboration from multiple departments, including pathology, our department, oncology, cancer predisposition, and subject experts. So the approach we use is what we call the 3 platform involving whole genome, whole exome, RNA-seq. So we actually started the work in 2013 to develop a computational pipeline that enable integration of the data from all these 3 platforms. In 2015, we launched the Genome for Kids protocol. This is when we start to analyze real patient data to look into the feasibility of applying these approaches to patients. 2017, we expanded the process to including rapid turnaround RNA-seq pipeline. This is to support the total 17 treatment protocol for evaluating target therapy for high-risk leukemia patients. And we also incorporated an FFPE pipeline that only include whole exome and RNA-seq in 2017. And in 2017, also we started to analyze the real-time clinical sequencing for every patient. And currently, we have already analyzed over 2,000 cases using the combination of the 3 platform rapid RNA-seq and FFPE protocol. So the computational pipeline for the 3 platform sequencing development was led by Michael Rush in my group. And so what we have done is to start with 3 platform sequencing, generating data, perform QC check, and then we have extensive analysis for both the somatic and sequence variants, including single nucleotide and copy number structural variation, as well as for germline based analysis. And then we perform variant classification using an algorithm we created called medal ceremony that include a harmonization of data resources coming from a variety of the databases that we have curated. And that way that we can perform assign gold, silver, and bronze medal to every variant we discovered. And then finally, a panel will convene to convert our medals into 5 class categories of pathogenic, likely pathogenic, variant of unknown significance, likely benign, and benign, to sign out the tumor and germline report for every patient's gone through this pipeline. So I want to mention that, with the applications of this approach to clinical sequencing, we actually can make therapeutic changes for a subset of the patients that are not enrolled in clinical trial. And this is one example that therapy changes occurred based on the use of the ClinGen data. And this is a patient enrolled in Genome for Kids, and he had the metastatic melanoma and who had failed multiple therapies, including the immunotherapy. So the tumor was analyzed by the 3-platform sequencing, and we discovered a MAP3K8 gene fusion for this patient. And the fusion involved loss of the last exon of MAP3K8. And based on literature search, that we found that this could cause auto activation because the last exon encode the auto-inhibitor domain of MAP3K8. So we project that this will lead to activation of MAP kinase signaling independent of BRAF. And the patient lacks BRAF V600E mutation, usually found in melanoma. So we proposed for using a MEK inhibitor, trametinib, and the patient got total response, but unfortunately developed resistance. So this is the case of N1 we discovered in our cohort. And the next question is that, do we found more patients with this fusion event? So in collaboration with Amita Bahrami at Pathology, who had an archive of FFP materials with spitzoid melanoma, we performed RNA-seq and discovered that MAP3A gene fusion or truncation actually has the highest mutational prevalence in this cohort. And these changes are mutually exclusive with all the other kinase activation events discovered in this cohort. Most strikingly, all these events involving the loss of the exon 9, as we can pinpoint the locations of these gene fusion or truncation event discovered in this cohort. So the pattern on the RNA sequencing data is also very clear in that we see a diminished exon 9 expression compared with exon 8. So then we were curious to see whether this event can also be found in adult melanoma. So the strategy we use is that we were able to design a very quick analysis, just looking at exon 8 expression to exon 9 expression differences on 472 melanoma samples, and was able to find 8 of them actually show a diminished exon 9 expression. So we just need to download these 8 samples and perform the RNA-seq gene fusion detection using the software Cicero that we recently published. And all 8 of them show gene fusion event similar to what we found in pediatric cancer. So currently we are collaborating with chemical biology department for testing new compounds that specifically targeting MAP3K8. And this study was published last year in Nature Medicine in collaboration with Amita Bahrami. Now I want to discuss lastly about some effort I really feel very passionate about, which is data sharing and visualization on Jude Cloud. So the study that we, the subject that we included in involves both the pediatric cancer patients as well as cancer survivors, as ensuring quality of the life of pediatric cancer patient survivor is also a main objective of how to develop less toxic treatment so that we can ensure there's a long time, high quality life of the survivors. We also have 800 pediatric sickle cell disease patients uploaded in the Jude Cloud. And this work was led by Clay McLeod and Xing Zhou in our department. So why doing the cloud platform? What we believe is that the traditional approach that we have, which involves a data repository that will require every users to download their own copy of the data and set up our own local infrastructure to perform the data analysis. While doing the cloud-based platform, we only need to upload the data and the tools once, and everyone can use the shared computing infrastructure on the cloud to perform this analysis. So we think in the long term, it will be the most efficient use of the data and tools that we developed. So Jude Cloud actually, as I mentioned, including data from both retrospective research study as well as from prospective clinical sequencing initiative from St. Jude. This is a project I introduced briefly before. So the data set we include involving pediatric cancer patients, about 3,000 of them now, cancer survivorship, about 7,000, and non-cancerous sickle cell disease, 800. So we perform data harmonization, curation, metadata collection, and also the data will be kind of shared around 3 different platforms. One is a Genomics Platform. This is a platform that allows users to be able to access raw data, run analysis. And I want to mention this platform was developed in collaboration with DNA Nexus, which can ensure safe cloud-based genomic data storage, and also Microsoft Azure. And I want to really mention that Microsoft actually played a very important role in our endeavor, ensuring that we get free storage and computing on the Azure platform when developing this infrastructure. And another part is we call the pediatric cancer knowledge base, where we have called PCON, where we have obtained published data and curated data from PCGP and other studies, so that the research community can freely access these information without going to curating data from different resources. And data visualization tools, it's called visualization community, that enable users to interactively explore these data. So the PCON and the visualization community can be accessed without applying for data access, while the genomic platform, we require data access agreement, as we want to ensure the patient data are protected with security. So currently, Jude Cloud have about 1.2 petabytes of raw data from over 10,000 patients. Over 200 data access has been granted for raw data, and we have close to 2,000 registered users that they can perform on-the-fly analysis using the cloud infrastructure. And per week, we have 2,500 unique users. And our last month, we actually exceeded the 10,000 user marker on the monthly basis. So this is kind of the breakdown of the retrospective studies and the prospective clinical sequencing data that currently hosted on St. Jude. And I just provided the paratumor normal genome and survivor whole genome tumor subtypes here, so we can see that we are able to represent major subtypes of pediatric cancer dataset on St. Jude Cloud. So I want to mention a little bit more on our real-time clinical genomics streaming. And this is an effort that we have put together to ensure that research community can access data as soon as possible without waiting for us to publish the first papers utilizing these data sets. So we call these RTCG. And the RTCG pipeline actually was developed so that the ClinGen data can be uploaded onto St. Jude Cloud. So it involved a couple of very critical steps, including verifying the patient consent. And this verification actually was done on a regular basis because we also need to verify there's no retraction of the consent, and the retraction happens, we'll pull out the data set of the St. Jude Cloud to ensure it's in compliance with the current status of the consent. The other one is extensive QC check that went in, and also the metadata collection. And we also performed the data harmonization to ensure that the ClinGen data are in sync with what data has already been uploaded on St. Jude Cloud. So I want to mention that this is also almost like a semi-automated process that was launched in May 2019. And these are the data points reflecting the data growth from March to July this year. So this is the pandemic hits, and we were able to see that even with the with the COVID affecting many other things that we're doing, that at least our data sharing effort has not been hampered by the virus. So rare cancer types is always difficult to assemble, as the real-time clinical sequencing analyzes every eligible patients in St. Jude. We're able to see that some of the very rare cancer subtypes are represented in our data offering on St. Jude Cloud. So availability of these data sets will really help with gaining knowledge about how to treat these rare cancer subtypes in the future. So we don't want data just sit idle on the cloud. The whole purpose is to ensure that researchers can also use some of the innovative tools that we have uploaded on St. Jude Cloud to do the analysis. And our approach is to ensure that these tools was designed in a way that by point and click, the people can utilize our tools and have nice visualization to look at the result. So currently the tools we have put on the cloud, including some of the tools that were wrapped around with the very popular algorithms that are used by everyone, as well as some of the new tools that we published, the researchers, faculty members in computational biology in my department has published. So I want to highlight one particular tool called cis-X. This is a tool that was developed in my laboratory by one of the former postdoc fellow Yu Liu, with the goal is to discover regulatory non-coding variant in individual cancer genomes by using this tool. So it was published only 2 months ago. So the design of the software is that you can actually input samples with paired tumor normal whole genome sequencing and RNA sequencing data. So we're utilizing a pattern called allele-specific expression and outlier high expression, so that we can look into cis-activated variants in that as a signal for non-coding driver variant. So the approach that we do is that you don't need to assemble a huge cohort for doing these type of the non-regulatory variants, and you can utilize individual cancer genome to find out these driver variant to do the analysis. And we also look into incorporating some of the additional features like transcription factor binding sites, and looking for utilizing the gene expression matrix we have assembled through St. Jude Cloud, and also looking at the TAD domains, others to find the potential candidate of these. So we performed a benchmark analysis using T-lineage AL samples because TAL has many of the known kind of non-coding variants, and they actually were researchers published through at least last 5 years. Many of the studies publish these non-coding variants in this disease. And by using cis-X, we were able to discover nearly all of these events and also find some of the new events like LM3 activation, 3 enhancer hijacking, and the tau1 activation through an intronic variant that hasn't been not being reported before. We also discovered a new potential oncogene called PRLR, and this variant, I will show a little bit detail, is discovered through the process of looking at the variant from whole genomes that are heterozygous. As you can see, the two colors representing red and the blue here. But in RNA-seq, only one color was shown, indicating only one chromosome was activated. And we are able to see there is a deletion event leading to the removal of CTSCF boundary elements from the existing data, suggesting that enhancer elements involved in IL-7R was interacting with the PRLR. This is a gene involved in JAK signaling pathway, and the deletion will enable interaction of enhancers over here by that. And we were also able to show that even though the event was only discovered in one case in our Shanghai cohort, that we are able to replicate the findings in two COG samples that were analyzed by NCI target. So now, the cis-X program is available to anyone who wants to utilize this, the analysis pipeline to perform, to analyze their own data. So I want to mention is that switch gear about the pediatric cancer knowledge base, and this is something that what we want to collect is to compile the curated variants from both the St. Jude data as well as the NCI target cohort, as well as other major institutions who are involved in research in pediatric cancer. So we have a close collaboration with Germany, and we are able to incorporate the samples they analyzed for their study into the PCON as well. So I want to mention that, for example, the TP53 gene locus that we are able to incorporate data from, 3 published studies, and also have an approach of showing both the sequence mutation as well as expression changes in parallel for these type of investigation. So the sequence mutations we include those that identified at the diagnosis as well as those found at the relapse. And I want to point it out that in this P53, there's the R248Q mutation that we can see. It is not only a hotspot at the diagnostic samples, but also were present in the relapse at the hotspot, which is not found in any of the other hotspot mutation, suggesting there's some unique mutagenesis mechanism leading to our this hotspot mutation. And we are currently investigating this in collaboration with our Shanghai colleagues doing the cell line model. So the visualization community, a new addition of this is the Tisney plot that we performed in looking into organizing the data sets, RNA-seq data set we have that enable classification of patient samples by expression profile analysis. So one of the new interface we added is that not only we analyze our own data, but also allow researchers to import their own RNA-seq data and see where their sequencing, their data sample can be clustered with the reference cohort we assembled here. And we are already using that for our own internal clinical sequencing effort to help with classifying some of the hard to diagnose samples that we encounter. So the visualization community is not as, not only taking data from de novo, but we also trying to incorporate the graphs that has been published in variety of pediatric cancer researchers into interactive visualization modules that people can explore. So this is a screenshot of the pan-neuroblastoma studies that we published recently on Nature Communication that involving analyzing neuroblastoma samples into that were using data generated from 3 different institutes. So this graph actually is visualization is actually interactive, and that can be explored in our visualization community as a dynamic graph. So if people here are interested in neuroblastoma, I highly recommend you to check it out on St. Jude Cloud. So the future and the ongoing work that we have, one is we also have recently developed a new tool called Genome Paint for visualizing non-coding variants, so that some of the epigenetic profiling data, including Hi-C, ATAC-seq, Capture-seq, can be utilized to identify driver variant. The example here showing is a duplication event of enhancer region that we found in bilineage AL that interacted with c-MYC promoter. So this study was has been accepted in Cancer Cell, and the visualization tool is already accessible to anyone interested in it, and you can find that on St. Jude Cloud. We also show that we actually, using this tool, we discover something that may unveil some of the novel mechanisms that affect the gene regulation in pediatric cancer. So over here, working with Mark Valentine at St. Jude, he designed RNA, performed a sequential RNA DNA fish experiment, showing that the RNA enhancer expression is only occur in the amplified allele. More importantly, these the duplication actually lead to the formation of RNA cloud, suggesting the enhancer RNA may change the chromatin structure that affects the 3D genome architecture of on St. Jude on the c-MYC regulation. So we also actually working with NCI right now to port protein paint to NCI genome data commons. And they found our tools can be only used for pediatric cancer, but could also help with advancing the visualization of adult cancer. So we are also looking into support for non-cancer genome analysis, utilizing some of our variant pathogenicity calls. So for example, working with Marcin Brodowski on looking at the MDS germline variant analysis. And we are very interested in evaluating the possibility of hosting data by external collaborators. So this is the end of my talk, and thank you very much, and I'm ready for questions.
Thank you so much. That was a great talk. I really appreciate it. Questions are starting to come in, and we're going to jump right in. First question came from Lauren Mills, and her question is, what level of intermediate data are available? Can we get access to mature SNP, CNV, fusion, and expression data?
Yeah, so the intermediate data included all the curated somatic mutations that we used for, curated meaning that those that's involving studies that were published. So for those data, somatic, this is genome-wide SNV indel and structural variation data that we have recently uploaded on the cloud. We have also offered RNA-seq feature counts to users who wants to use them. You don't need to apply for data access for these. So for, in terms of a gene fusion event, just a couple weeks ago, we made an effort trying to what we called annotate subtype biomarkers, meaning that gene fusions are sequence mutations that define a specific pediatric cancer subtypes. So for example, if people wants to use analyze rhabdomyosarcoma fusion positive versus negative samples, they don't need to re-download and reanalyze everything, but they will be able to, just using the annotation we provided on the sample metafiles to categorize this information. So we'd be very interested in people to actually utilizing the data, give us more feedback on what are the additional data elements that we can provide to serve the research community.
So the next question comes in from Yang Ding, and it said, thank you for the fantastic talk. Really wonderful to get an overview of the exciting tools and work you guys have done and inspiring for young investigators. And then in your variant calling pipeline, what tool do you use to call somatic structural variants?
Yeah, so the somatic structural variation that we use is using the cis-X algorithm that we have developed. In my lab, developed very early on 2010. So we use that in conjunction with another tool called Concerting that is a copy number variation tool. So because what we found is that if you use so for some of subclonal structural variations, you will end up not having the power for detecting the subclonal structural variation if we just apply CREST alone. We do recognize there's, CREST was developed almost 10 years ago. The new modern tools are available. So what we have recently did a fair amount of the benchmark analysis, and we found that Manta appeared to be a good software that is complementary to CREST in terms of can discover additional variants that were missed by CREST. But there's also a subset of the variant Manta cannot discover, but CREST can discover. So we do plan to run Manta on all the cohorts we host on St. Jude and provide the results to the community. So for gene fusion event, we use the Cicero algorithm that recently published in Genome Biology, developed by my own lab for this endeavor. And we're also looking into some of the other softwares like SV-ABA to perform a potentially kind of making sure that there's no missing event in the data set we have.
Thank you. The next question comes from Barbara Rivera, and she said, amazing work, thank you so much. Would it be possible to submit their data for analysis? How would we need to work around ethics on that sense when submitting data that requires controlled access?
Submitting data require what?
She wants to know if it's possible for them to submit their data for analysis on your platform.
Yeah, if just submitting for analysis alone, you don't need to have any, you just upload your data on DNA Nexus. They provide the security, and you can just use our tools to perform the analysis. But if you want to use us to host your data on Jude Cloud, that we would like to make sure there is agreement that we can have permission to, like data hosting agreement from you. And the data access actually will be approved by the researcher, not us. So the service we'll provide is to ensure there's a data harmonization that happens so that the data that's supported by, provided by the user could be harmonized with the existing infrastructures that we, existing data sets that we have. So we're very open to both supporting the data analysis and also data analysis as well as data sharing, utilizing our infrastructure. And also, I want to mention last 2 weeks ago, I was at UCSD, and I was really thrilled to hear that researchers there have performed the episome analysis using one of their very innovative algorithms. They just uploaded their tool to the DNA Nexus platform and was able to get just analyze the a subset of the brain tumor samples that they are interested directly utilizing the data we provided on the cloud. So this is kind of really when I'm really eager to hope that especially computer scientists who are craving for data, this is a large resource that you can get access to, no obligation for collaboration at all. That we would really like to share this data to advance the pediatric cancer research as a whole.
Thank you. And she responded and said, thank you, she understands now. You have time for a couple more?
Yeah.
Okay. Next question is from Payal Jain. Thank you for the fantastic talk and your team's work. My question is, if you observe discrepancies in mutation calls between WGS and WXS for the same patient tumor, is that likely due to different read depths, caller metrics, et cetera? And then how is that handled and which data modality is prioritized, both on research and the clinical end?
Yeah, so I will kind of, talk about the clinical part, because clinical is where we did the whole genome and the whole exome sequencing. So this subject actually has been described in detail in our Nature Communication paper, the Rush et al. Paper, that we have shown examples of how we resolve these. So what we have done is that, for the clinical, because everything is treated with, we have to report variant to sign out reports. So any discrepancy was evaluated manually by a scientist. So what we have learned is that, yes, there is, for variant that was discovered only by whole genome sequencing alone, this may be usually related to the capture efficiency of exome. So when you have for example, an indel coupled with, the polymorphisms in the exome that were used for capture, you actually have less efficiency of capturing the indels in exome sequencing. That will lead to a missed call in exome compared with whole genome. So many times we can actually use RNA-seq data to cross-validate one of these the platforms. But if the variant is not expressed, then you're out of luck. And then because we have developed a lot of visualization tools, so it's eventually the curator is going to make the final call on that. So for sequence variant that were missed by whole genome but found in exome, these are mostly related to the subclonal variant, that the whole genome data, which we are now using 45x coverage, may lack the power for discovering them.
Thank you. So last question is going to come from me. At first, I want to thank you for that neuroblastoma slide. I appreciate that. You know that we care a lot about neuroblastoma. I was really interested in the three platform sequencing initiative that you're doing. And it seems like you guys are investing a lot of money into that. And do you know what the cost you're investing per patient to get all that done?
Yeah, so I think the cost has a lot to do with the, with the platform. So when we started, it is very expensive because at the time we don't have HiSeq. So that cost about $8,000 sequencing cost per patient. With the NovaSeq, which we're using in clinical right now, it is $3,000 a patient. I think the people always talk about sequencing cost. How much does it worth the effort? I felt you have to look beyond the value of this for just signing out the clinical report, because as you could see that as we share the research data within St. Jude and also with the broad community, the value of this is go beyond just the signing out the patient samples period, but also to enable research to be carried out on these samples. So we already published 8 papers that utilize these clinical sequencing data for a variety of the investigations. And also, I just want looking at the data access request for our that we received, that more than 50% of the data requests are actually targeting the clinical sequencing samples. And these are the most beautiful and highest quality data we ever generated, because these machines were dedicated to clinical sequencing. There's no contamination. So even for just something like interesting, like for example, we also were looking at the virus, is any viral genome integration in pediatric cancer patients, right? Virus analysis. When we look at the data generated from research cohort, so these were from the Washington University, the whole genome sequencing were done in Washington University, Hudson Alfred, the reference laboratories. we actually pick up viruses not supposed to belong to the human genome, like viruses in plants and stuff. So it indicating there's some tiny, because the same sequencing machine were used, you kind of picking up the residual, these effects of machines being used for other samples. But for the ClinGen, as these machines were dedicated for the use of patient samples, no other viruses except the human being detected that. So I believe our ClinGen sample is the most beautiful and the highest quality cancer genomic data ever existed. Any computational scientist want to use for developing new tools, that's the best data set you can get.
Yeah, so 3,000, I mean, it's a one-time cost. Once you do it, you have it.
Yeah, once you do, you have it. You can mine it for other reasons. You can do things you never thought about possible for doing. So, for example, germline variation interpretation, right? So sometimes it's hard to tell because, for example, we think if it's a variant is involved in some DNA mismatch repair, is this real or not real? If you want to have one copy, does it matter? So with the whole genome sequencing data, we're able to utilize in the data, like perform mutational signature analysis. Because pediatric cancer has a very low mutational burden. You cannot run exome for mutational signature analysis, but whole genome, you have enough power. And then we detect DNA mismatch repair signatures that actually can have an implication for therapy in the future.
Wow. Thank you. Thank you for answering that. And thank you so much for your time and your great talk. We really appreciate it. We appreciate everything that you're doing, and hope that you stay well and be safe out there.
Thank you, Jay. Very nice to participate, and thank you for the opportunity.
Okay, take care.
Bye.
Bye-bye.